really excited to talk to everybody today. Wanted to thank Cure HHT Canada for giving me the opportunity to talk to you guys about HHT science. My first patient conference was in Boston, and I think it was in 2016, and I heard a similar talk that I'm about to give you today, and I just remember being really inspired by it. So hopefully I can do the same for you guys and give you a pretty decent overview of where we're at with HHT science. So just to give you an overview of my talk, I'll talk about HHT background and the circulatory system. I know you've heard it quite a bit today, but repetition is key. And then we'll get into kind of the causes of HHT, as well as, you know, the different signaling pathways and really what does the signaling pathway mean. And then I'd like to discuss some models that we use to study HHT, the different cell models, all the way to animal models and to even human models at this point. And then I'll briefly discuss some of the recent scientific advances we've made, both in genetics and cell biology. And most importantly, why research is important, why basic science research is important, and what you guys can do as patients and individuals to help. All right, so I won't belabor this too much, but as you guys know, HHT is an autosomal dominant vascular disorder, which is characterized by the formation of arteriovenous malformations, which are direct connections between the arteries and the veins, completely bypassing the capillaries here. What's really interesting is that the prevalence is at least 1 in 5,000 people, but it's thought to be highly underdiagnosed. And this is likely because the symptoms from one patient to another are very different, and even the onset of those symptoms can be very different. What I think is most interesting is that it's estimated that 90% of people are currently undiagnosed. And if that's the case, then the disease prevalence is actually 1 in 500, which is not a rare disease. And I want to emphasize that because I try not to say rare disease because I don't think it's that rare. I think a lot of people are affected by it and a lot of people should care about it. But despite it being highly underdiagnosed, the diagnosis criterion is really quite simple, right? So, you know, you have your epistaxis or your frequent nosebleeds. You have the different skin and mucosal telangiectasias, the different visceral AVMs, which are those larger AVMs that can occur in all different types of vascular beds. And then, of course, a family history as well. And so I'll show this to you guys, and I won't talk about it too much, but I just want to briefly mention or emphasize the fact that, you know, we have these AVMs that take many different types of forms depending on what sort of vascular bed they're in, and really they look very different from one patient to another. So no AVM is alike. And so this is one of the things that, at least in the field of science, that we're really interested in, is understanding why certain AVMs look one way in one vascular bed and why they look very different in another vascular bed, and why they have different disease outcomes. But to really understand HHT, we first need to have a basic understanding of the circulatory system. So what is the circulatory system? Well, the circulatory system is composed of your heart, which beats blood, which pumps blood through all these different blood vessels here. And so this is just another way to show it here. So what's really important about carrying all this blood to all these different types, or all these different areas of your body, is that it's really important for your body to receive the proper oxygen and nutrients for it to be healthy. And so just to give you an idea of what happens here, you have blood on the right side of your heart here, which is pumped out into your lungs, and where your lungs will oxygenate this blood. This blood will come back into the left side of your heart and then pump into the rest of your body to deliver that oxygen and those nutrients to all those vital organs, including your brain and your liver, your intestines, and that sort of thing, and even your muscles. The one thing that I want to emphasize too is just how interesting the range and size of your blood vessels is really quite grand. So I like to use the example that the blood vessel kind of looks like a tree to me. So you start with this really large vessel that branches into smaller vessels, that branches into even smaller vessels, that branches into tiny vessels. And to give you an idea of just the magnitude of the size difference, is that When you look at the aorta, which is one of the largest vessels that you have, it's about 2.5 centimeters wide. And that's roughly as wide as like a highlighter or a really large marker. And as you go down and branch down and get even smaller and smaller, you begin to have these capillaries, which are only 5 microns in width, which is essentially the width of a spider web silk. So these are really tiny vessels that are carrying blood through your body. And I mention this because it's really important in HHT. You know, when we look at a regular vascular bed, we can see that this normal vascular bed branches and you get these really nice small vessels. And these small vessels are really important for two specific reasons. The first reason is that these small vessels are really good about distributing oxygen and nutrients to all of the tissue. And the other thing that they do is that they take the pressure, they relieve pressure across these blood vessels. So the pressure here is going to be much lower than it is going to be here. And so when we think about HHT and AVMs, we have this really large arteriovenous malformation. So this is going to be a high flow, really high pressured system, which is likely going to induce an hemorrhage. And you also don't get that good distribution of the oxygen and nutrients. So you can see why this could be a really big issue. Now, when we talk about the blood vessels, we want to know, okay, what's the building blocks of the blood vessels? And those building blocks are cells. And these cells are called endothelial cells. So this is a picture that I took in the lab not too long ago. This is a whole bunch of human endothelial cells that are cultured in a dish on a 2D surface. And you can see they have really nice long architecture. They have these really, these blue circles here are the nuclei of the cell. The nuclei of the cell contains all the genetic information. And so when we look at it in the sense of a blood vessel, let's say we had a blood vessel and we take a cross section of that blood vessel. And the first thing that you notice is that you have this really wide lumen here. So this lumen is empty space where the blood will flow through. And I want you to appreciate how strong these vessels basically have to be to contain all of this blood that's running through this lumen. And so these are your endothelial cells here in red. And these endothelial cells are basically reaching over to one another to make a nice seal so that nothing leaks out, or at least not too much blood leaks out. And then these blue things here, again, are the same blue things that you see in here, which are the cell nuclei, which hold your genetic information. And that's important because when we talk about, you know, what is HHT and how it affects our bodies, well, HHT is really a disease that affects our endothelial cells, and which is what you see here. And so when we talk about what really is happening at the molecular level, we can look at, you know, what's causing HHT, which is these genetic mutations which occur in this single signaling pathway. And I'll explain this signaling pathway in greater detail, but what I want you to really focus on right now is what you've heard of quite a few times, is that there's really 3 main genes that are mutated at HHT. The first one is endoglin, which is codes for this protein endoglin, and this is a receptor that sits on top of the cell. You also have ACVRL1, which encodes for the protein ELK1, which is another receptor that transmits signals into the cell. And then the third gene is going to be SMAD4, which is HHT-like syndrome of juvenile polyposis. And this is essentially a really important protein that, once it's activated, will go into the nucleus and it'll read the DNA that you in each of your cells. And so one important thing to note too is that, as Samantha probably had told you, that 95% of patients will have either an endoglin mutation or an ELK1 mutation, and the remaining 5% will likely have a SMAD4 mutation. So really a high probability of these HHT patients will have mutations in these 2 receptors that are on the surface. And so I'm saying receptors and I'm saying all these proteins, but really what does all this mean? So really, what is a signaling pathway? And when we talk about a signaling pathway, hopefully you can appreciate it in this sense. So we have our cell here, and you can treat our cell like it's a factory. And this factory makes clothing. Let's say it makes any type of clothing that you want. And this factory has this radio tower that sits on top of it that receives any sort of signals from the outside world. And so let's say the outside world is saying, oh, we have a really big blue gene shortage, and the pandemic was really, really bad to us. So we have a shortage in just about everything. And one of the things that we need the most is blue genes. And so there's a message that gets sent that, hey, we got a blue gene shortage. And that message is then received by this radio tower. Once it's received by the radio tower, it will transmit the message to Bob, who's basically in charge of the whole factory. And Bob will get his workers on board and will ask Cheryl, hey, get your guys to start making more blue genes because there's a blue gene shortage. And then Cheryl will go to Jim and say, hey, Jim, start that machine so that we can start making more blue genes. And then Jim will start the machine, and then you get more blue genes. And so, okay, well, what does this mean with respect to HHT? Well, if you think about the message, which is this gene shortage, this is like our ligand. So these are the proteins that will send a message to our radio tower, which at our radio tower here will be our receptors, ALK1 and endoglin. So the message will get received by the receptors, and then the receptors will transmit signals to different workers or proteins. And these proteins will then get activated, and then Jim here, which would be SMAD4, that protein will go to the nucleus, and it will turn on the machine to have the machine read the DNA and have some sort of gene expression. And so when we think about this signaling pathway and we think about HHT, one of the things that comes to mind is, well, okay, we know that this pathway exists, but we have no idea really, we still don't really have a good idea of what it's doing. And so to be able to understand what it's doing, we have to have specific models that we use to study the disease. And so these models start really simple. They start as using endothelial cells or different blood vessel cells that we just look at in static cultures. We can then apply blood flow to them to simulate blood flow over those cells. Or if you want to get a little bit more physiological, we can take these cells and we can put them in 3D matrices, different 3D matrices, so that they can start to grow blood vessels. And then, of course, if we want to become more physiological, we can use different animal model systems like mice and zebrafish. And then, of course, humans is really the ultimate model that we want to study. And so just to give you an idea of each of these models, if we first start, how do we simulate HHT in these cultured cells? And so these are isolated human endothelial cells. So these are blood vessel cells. And we can actually take these cells and we can knock out or delete different HHT genes in these cells, like ACE4L1, endoglin, or SMAD4, and then study their cell shape and their behavior and all of the different molecular mechanisms that we can see. But if we want to simulate blood flow, the way that we do that to make it a little bit more physiologically relevant is this is one of our microfluidic devices that we have. So it's a really beautiful device, but anything in orange here that you see is where the cells are going to be basically laying on this glass cover slip inside of this chamber. And then to simulate blood flow, just like the heart, we have this peristaltic pump, which will pump media or blood through this mesofluidic device, the microfluidic device, and basically simulate blood flowing over these endothelial cells. Of course, this is a 2D model, and endothelial cells exist in a 3D environment as blood vessels. And so if we want to study this in a 3D aspect, we can do this with the help of Chris Hughes from UC Irvine. He's been able to miraculously generate blood vessels in a dish. And this is a really cool model because not only do we get this 3D architecture of the blood vessels, but we can also study the molecular mechanisms at the same time with very high resolution. So we can really see everything that's going on. And the amazing thing is that he's been able to create AVMs in a dish. So he can knock out or delete these HHT genes in these cells seed these cells in this device, and they can form these blood vessels, and they can actually successfully generate AVMs in this dish. And I'll play these. So these are fluorescent beads that are flowing through this AVM right here that you can see through from this side to this side. So they've done a really, really cool job in being able to create just a really fantastic model for looking at AVM disease mechanisms, but really for being able to perform really high throughput drug screening. So you can take this AVM in a dish and you can test, hundreds of drugs all at the same time and see if they can reverse these AVMs. And so going in accordance with this, people have started using what's called human stem cells, or iPSCs. So the way that scientists do this is they basically take a skin biopsy from a human. They take their skin cell, they basically induce it to become a stem cell. So they de-differentiate it back into a stem cell. And then using that stem cell, they'll then differentiate it into whatever cell that they're interested in. And of course, for us, we're interested in blood vessel cells. And this is a really powerful tool because we can actually take cells from HHT patients who have all different kinds of, HHT mutations or genetic mutations, and we can test different drugs. We can test how their disease is different from another person, and we can develop therapies based on that. So it's really the forefront of individualized medicine. And so coming from this cell culture model systems, we often really want to see if everything that's happening here is also happening in these higher level organisms. And so the first thing that I want to introduce to you is zebrafish as a model for HHT. So we use zebrafish extensively for HHT, and they're really great because they're super small, easy to handle. They produce a high number of embryos, so we can study a lot of embryos at the same time. They're really easy to manipulate on a genetic and physical level. So we can induce mutations really quite easily in specific parts of the different genes that are responsible for HHT. And they also develop very rapidly. So we can do this really quite quickly. But most importantly, they're optically clear. So this is a really nice picture of a fluorescently labeled blood vessels in this zebrafish. And I say that because it's really good that they're optically clear because we can actually see AVMs develop in real time, which has been really important in understanding the cellular mechanisms of AVM development. And so we have the ACE401 mutant fish, and there's also Arndt Siekman from University of Pennsylvania, who also has endoglin mutation mutant fish that we use currently to study the disease. And just to give you an idea of what the AVM looks like in a fish, is if you look from like the top down, this is their head right here, and this is their cranial vasculature. Hopefully you can appreciate that these red vessels are going to be your arteries, and the blue vessels are your veins. And so you have really nice architecture here. But in our mutants, the ones that we delete ACE401 from, when we get rid of ELK1, we can see that obviously you see that these vessels get really large, but most importantly, they develop an AVM. So they develop this direct connection between the artery and the vein. And so again, this gives us a really good tool to study AVM development in the fish. Of course, a lot of other labs use mouse models, and mice are really unique, and they're really strong models because they have very similar physiology to humans. Obviously, with fish, they live in water and they breathe oxygen from the water, but they don't breathe air, where mice, they certainly breathe air. They're very easy to use, and to study the pathophysiology of HHT, you can look at all different kinds of vascular beds, like the retinal vasculature in the eye, the cranial vasculature, the intestinal vasculature. And there's 3 different mouse models that are currently being used. There's the ACE401, the endoglin knockout models, and those are from Paulo and Ian Eichmann's lab from Arizona and Yale, respectively. And then you have the SMAD4 knockout model, which is used by Strider Meadows and Roxana Ola from University of Tulane and Germany as well. And so just to give you an idea of what this looks like, if we look at, this is a retinal vasculature of a mouse, and it's really quite beautiful, the kind of the morphology and the shape, the architecture of the vasculature. So in the control, you have pretty normal. When you knock out ALK1, hopefully you can see that there's this AVM right here. And if we blow it up, you can see the artery here that turns into the AVM right back to the vein. And so you have that direct connection. And similarly, we see the same thing when we look at endoglin knockouts as well. The control are normal, but then you see hopefully this really big swooping blood vessel, that's the AVM. And of course, when you blow that up, here's the artery, and that's the AVM right there that goes back to the vein. And what Paul O actually has been able to do is he's been able to locally induce AVMs in the brain vasculature, which has been really great, because prior to this, people really only were able to study the retinal vasculature. So to be able to make it organ specific has been a really powerful tool. So they delete ACE1 in these mice, and they look at, the really cool thing is that they're able to look at AVM development over time using MRI. So this is the MRI image here that you can see. And this isn't published yet, but they can actually control where these AVMs form by deleting the HHT genes in specific areas of the brain. So you can really get a good idea, you know, if you do it more in the outer cortex, what happens when you have an AVM there versus more in the midbrain, or like near the hypothalamus, or any of those really important areas for memory. So obviously we have our animal models, but what about human models? So probably fortunately, we don't do very much human work because most humans don't like when they have experiments done to them. But what we have been able to do is we've been able to do genetic sequencing of human cells from isolated telangiectasias. And really this has been pioneered through the Doug Marchuk lab out of Duke. And so essentially they're able to take these telangiectasias, do a biopsy, they grind up that tissue, and then they extract out the DNA and they read the DNA to basically look at all the different types of mutations that can occur. So it really kind of lends itself into really looking at the genetic code of, you know, individualized patients in these individual telangiectasias. So you can become really specific about it. So we have all these models of HHT, but how do we use these models to attack HHT scientifically? And really the goal of it is to understand the underlying mechanisms that lead to disease. So what's happening on a molecular level that induces the AVM? And so when we think about, when we try to understand these mechanisms, we can leverage this knowledge to basically generate new technologies and new therapies. And so when we think about how to understand the mechanisms behind the disease, well, we have to, again, go back to the signaling pathway and how it affects cellular behavior. And so one aspect of attack for looking at HHT scientifically is to study the ALK1 ligands. And these are those messages, the BMP9 and BMP10. These are the messages that will basically get received by that radio tower or the receptors, right? And so when we look at, we've been able to do a lot of work here at Pitt in the Roman lab and as well in collaboration with Andrew Hink in understanding the structure of these ligands and how they're processed or secreted into blood, which is really important because if we understand the structure, we understand how they attach to ALK1 and endoglin. We've also been able to find out that BMP10 is really the required ligand for this signaling pathway. And this has been confirmed by work done by the Paul O lab in mice as well. So it seems like, you know, we have these two different messengers, and it seems like you really need BMP10 in order to maintain good vasculature. Although there's been, of course, always controversy with regards to that. And of course, again, knowing and understanding the structure of the ligands, we can develop different synthetic ligands to help stimulate ALK1 receptor at an even greater level. So, if we don't have ALK1, in that we're not getting as much ALK1 signaling, can we develop a synthetic ligand that will really attach to ALK1 and really signal at a great effect? So that's one point of attack that we can use. The other point of attack is really studying the receptor. And so the first thing is studying receptor expression, which is essentially the presence of the receptor. And so work again done from Paulo has looked at if they increase the amount of ALK1 that's at the surface of the cell, they actually can rescue mice from endoglin deletion. So these mice that develop AVMs through an endoglin deletion, if you overexpress ALK1, then you don't have any AVMs. Similarly, a lot of the work that I've been doing is to really understand how to increase the levels of the ALK1 receptor, at a natural level. So how can you get the cell to do it on its own? And I've seen that blood flow is really essential for that process. And so there's still a lot of work ongoing for that. And of course, all of this trying to understand, if we understand how these receptors are expressed and how they become present, can we develop drugs that will enhance their presence and enhance their ability to signal? Of course, there's also studying the receptor function. So what's really happening at the back end when you stimulate the receptor? What happens with endothelial cell behavior? How does the cell behave in such a way that leads to AVM formation? And so a lot of work has been done both in our lab and both in Ann Eichmann's lab as well, as looking at the loss of ALK1 and endoglin, which will result in defective endothelial cell migration. So what does that mean? So these endothelial cells that make up the blood vessel are actually really motile, so they move quite a bit. And we've seen that in normal setting that these cells will actually migrate against the direction of flow. So they'll move against the direction of flow. But when we delete ALK1, we see that those cells actually migrate back with flow, and that aids in the development of the AVM. The other thing that we've noticed too, and that Arndt Siekman has noticed, is that the loss of ALK1 or endoglin actually results in cells becoming very enlarged. So when you lose ALK1 or endoglin, those cells become really large, which makes the lumen of that blood vessel really large and induces that AVM. And so, of course, the next kind of step is to understand, well, what's happening to these cells that makes them migrate in the opposite direction and makes them get really big. Of course, the other point of attack is looking at the genetic level. And so understanding what genes are regulated by ALK1 and endoglin signaling. And so there's been a lot of work done over the years to try to understand this. And it seems like that ALK1 signaling appears to act as a brake for overgrowth of blood vessels. So during development, you have a whole bunch of signals that are happening that are telling blood vessels to grow. And ALK1 seems to be there so that it can put a damper on that and say, hey, let's not grow too much so that we don't grow any AVMs. And so that really has been based off of a lot of the drugs that have been in clinical trials right now, which are called anti-angiogenic drugs. These are the same drugs that are trying to inhibit those signals that's telling the vasculature to grow, to continue to grow. And of course, our very own Anthony Canavici has been looking at dysregulated microRNAs and how they play a role in HHT. So if you look at this picture here, you see these little small things here. These are little pieces of genetic material that actually bind to the DNA or RNA or what have you, but they bind to other genetic material and they can actually stop certain genes from being expressed or being present, or really several other different functions. But these are kind of a lot of the things that have been coming out, at least on the genetic level lately. Of course, when we think about our genes, we think about gene editing. Well, can we just fix the genetic mutations. And there's really been a lot of work that's been done using gene editing techniques like CRISPR-Cas9. And this process is essentially, you have a piece of DNA that's mutated, and you want to cut that DNA out, and you want to put the right DNA back in, right? And so you use a protein that is saying that will identify that piece and say, hey, cut here, cut here. And then your Cas9 protein, which is your protein that cuts the DNA, sees that sign, gets recruited over, and then will cut the DNA out. Of course, this sounds really idealistic, but there's a lot of things that we still have to work through to make sure that this system is kind of foolproof, as we say. So of course, we want this to be very specific to the segment that we want to cut, right? We don't want to cut other DNA segments that may be really important. The second thing is that we often want to replace the DNA with the right sequence. And there's been a lot of new advancements that's allowed us to do that, but it's still a pretty complicated process depending on where inside the gene that you're trying to target. And of course, we want this to be targeted to the right cells. If you want to fix HHT, you don't want to cut the gene that's in the brain cells. You want to cut the gene that's in the blood vessel cells. So how do we target this to the right cells? And there's another thing that kind of complicates this, and it's kind of a complex idea. So I'm going to try to walk you through it pretty well. But work out of Doug Marchuk's lab by Dan Snelling has shown that HHT is caused by loss of heterozygosity. So, of course, what does that mean? So, as Samantha told you, all of humans have heterozygous mutations. And so, what are heterozygous mutations? So, humans have two different alleles, one from mom and one from dad. And in HHT, typically one of those alleles is mutated. And if you have one mutated allele and one regular allele, that's what we call heterozygous. Well, in HHT, we found that there's a small percentage of cells within these telangiectasias or these AVMs that actually have some sort of second mutation to this other, that occurs at some point to this other allele, this second allele. And now they're not heterozygous anymore, they're homozygous. Both of their alleles has this mutation, and you basically don't get any sort of production of the protein. And of course, what does this mean for your cells if there's only a small percentage of cells? You get this phenomenon that we call ALK1 mosaicism. And basically what that means is that you have a certain percentage of cells that act normal because they're heterozygotes, so they have one normal allele. But then you have a certain percentage of cells that have a full mutation in both of the alleles. And so of course that kind of begs the question of like, okay, well, what fraction of these mutant endothelial cells are required for AVM formation, and how does this lead to? So these are a whole bunch of questions that we now have, and that we're now looking into to try to understand AVM formation at a even deeper level. And so when we think about all of what we've learned and all of what we still need to learn, really the big research priorities that we have in HHT is the first one is really to get FDA approved drugs for HHT. We need that to happen, and we need it to happen now. And there's a lot of clinical trials going on right now for several different drugs, and we're really hopeful that within the next few years that we'll have some FDA approved drugs for HHT. But of course, we still want to find more targeted therapies. And the only way that we can find more targeted therapies to the signaling pathway is to understand how these genes affect cellular behavior. So if we can understand how HHT genes affect, a specific cellular function, then we can fix that cellular function if the HHT gene isn't there. And then, of course, we need to improve our model systems. Right now, we don't have any model systems to look at pulmonary AVMs. We have the brain AVMs that I showed you, and we can also look at intestinal AVMs, but we don't have anything for the pulmonary AVMs. So that's really important because, like I said before, AVMs in one vessel bed can be very different in another organ or tissue. And so I think most importantly, what I want to talk to you about, though, is like, how can we make a difference in HHT as a patient? And the really important thing is participating in your clinical trials. Again, we have quite a few that are open now, and getting these trials completed and getting these drugs approved by the FDA. It's really important that we all come together and we all participate in these trials to see what drug's going to work best for everybody. Sorry. The other thing is to lobby to your government agencies to increase funding for basic science research. We've had really good success with this in the States. We've been able to open up lines of funding through the Department of Defense as well as the FDA. And I guess the other big thing too is that you shouldn't underestimate your donations. They go an incredibly long way. We've had in the States anyways, over a period of time, we've been able to give $2.14 million in seed grants. These are grants that are like $50,000 to $100,000 to young investigators so that they can collect preliminary data and they can leverage this to get these larger federal grants in the U.S. And so we've had about 2 million that turned into $51 million. So it's over a 2,000% return on investment. So really, again, your donations go a long way. So anything that you can do to support is just more than helpful. And then, of course, these different donations too can attract young researchers to join the fight in understanding this disease. I can tell you at our last scientific conference, we had a record number of attendees. We had, I think, over 300 people there. And what was even more amazing is that we had, I think, more than 35 young investigators that we funded through travel awards. And that was really only possible through the generous donations of patients and families like yourselves. And so with all that being said, I'd like to open the floor for any questions that you guys might have.
Yeah, thank you, Dr. Anzell, for a very informative and educational talk. I especially appreciated all the graphics and analogies like the blue gene analogy that really made some of these complex these complex concepts very easy to digest. And thank you to you for doing HHT research, and I hope you continue moving forward with that, and the best of luck to your projects. We do have a number of questions from the audience. So one question is, in HHT, do the mutations completely eliminate all protein production of each of the respective genes, or is it variable?
Yeah, so that's a really great question. So there's been a huge debate in the field of like whether or not, so if you have these heterozygous mutations, so one allele is affected, right? Sometimes there's proteins that are made, but they're not functional. Sometimes the level of protein is not nearly the same as if you were to have two normal alleles. I think the one important thing that we've learned, at least from our animal models, is that in our animal models, if we have these heterozygous mutations, so if we have one allele that is mutated and the other allele is okay, we actually don't see any sort of phenotype. We don't get AVMs. And it's only when we delete both of those alleles where we start to see the AVMs. And so I think that's why this loss of heterozygosity that I explained is really important, because it seems like you need a certain amount of cells that don't have any functional protein that to induce an AVM. Now, there's a lot of differences between animals and humans, and so this is really a really hot area of research that we're continuing to try to understand.
Great, thank you. Another question is, are there any other signaling pathways that can compensate for the mutations in HHT?
Yeah, that's a really good question. So there's been some papers that have looked at how ALK1 will cooperate with different pathways, like the Notch pathway, in limiting angiogenesis, or like being the brake to that blood vessel growth. But I think that's kind of what we're trying to figure out. So there's actually been a paper that was published by the Meadows lab that looked at angiopoietin 2. So it's like angiopoietin 2. Oh, that was actually inhibition. Yeah, sorry. So I guess with respect to your question, that's kind of the reason why we've been doing all these experiments to understand what's happening at the back end. So when ALK1 signaling happens, what does it activate? What other pathways does it activate? Because if we can figure that out, then we can attempt to enhance those other pathways, like you mentioned, to try to compensate for the disease. But at this point, yeah, we really only know how to inhibit specific pathways that we think are associated. I don't think we're at the point where we can enhance a pathway to compensate.
Thank you. Since HHT is a disease of the BMP9 and 10 pathways, are there already FDA-approved drugs that target that pathway that can be repurposed to treat HHT?
Yeah, so that's funny you mentioned that. So we actually have a lot of the drugs that are being tested for HHT have been FDA approved for other diseases, mostly for cancer. And I know, so BMP9 drugs are really important in another disease called pulmonary arterial hypertension, or PAH. I don't know offhand if some of the drugs that they're using are FDA approved or not for PAH, but I can't tell you for certain that a lot of the drugs that are in clinical trials right now are FDA approved for cancer, and they're being repurposed for HHT.
Great, thank you. Now, you covered quite a few animal models such as mouse and zebrafish, but are there any other types of animal models that exist, like monkeys or even pig models? And do you think they would have advantages over the current array of animal models?
Yeah, that's a good question. So there are tons of models out there. So there's and these are for different diseases that you can look at. So they have, like, you can use rabbits, you can use dogs, which are, not many people like the dog models, but you can use primates and that sort of thing, as well as pigs. As far as HHT is concerned, we haven't really been able to get to that point. One of the really nice things about mice and about zebrafish that we use is that it's really easy to manipulate their genetics. So we can give them the mutations for ACVR1 or endoglin or SMAD4. And some of these larger animal models, it's really hard to generate, different primates that or different animals that have these mutations, just because of there's a lot of protocols that you have to go through. And also, I'll say that mice and zebrafish, they give a lot of offspring, and so you have a much higher probability of getting the genetic mutation that you want in those specific model systems, which is why they're used. So yeah, there's not very many like bigger models, and it's purely just because the genetic manipulation is very difficult.
Thank you. So we have a few more questions. Can you comment on why certain tissues are more prone to AVM or telangiectasia development?
Yeah, so I can tell you that we really don't know, but I think we all have specific ideas. So Paul O did a lot of work looking at how injury can induce blood vessel formation and induce more AVMs. And so when we think about, like, the nose and what you heard in the last talk, when we talk about the nose, that the nose is really exposed to a lot of foreign material. It's exposed to a lot of injury, and so you're constantly getting, blood vessel growth, because you're getting turnover of those blood vessels, and likely that's why you're getting a lot of AVMs that are prone to rupture and that sort of thing. As far as, like, brain AVMs and pulmonary AVMs, I have specific ideas, at least with, like, pulmonary AVMs, and these aren't backed by any, like, sort of research, but it's just hypotheses. If you think about, like, a pulmonary AVM, those vessels are constantly being stretched out when you take every breath. So those vessels are being stretched out. So that has to have some sort of an effect. But I guess the overall factor is, the overall factor is if every single tissue is very different, it has a different environment, and that environment is going to have a profound effect on how blood vessels function and how they're formed. And just to give you one last example of like the same thing with your nose, your intestines are always constantly exposed to different food or toxins like alcohol or something like that. So that's just another area where you might have, even in your colon, like where you might have some sort of injury or trauma.
Great, thank you for your insights. There's one final question, and this one's a very broad question, so you may have some fun with it. But can you comment on the future of HHT research, and what areas of HHT research do you think should be the focus of more study?
Yeah, my research. No, I'm just kidding. So I think we're in a really exciting time when it comes to HHT research. Like I said before, our last scientific conference, we broke our record for the amount of, I think we had like double the amount of people that are coming. So more and more people are getting interested in HHT science. And that's really important because that means more people are working on the problem and we can get it solved quicker. As far as where I think the field of HHT research is headed, I think there's a couple different things that need to be prioritized, and I think they are being prioritized. So we had a meeting that looked at to kind of develop like 7 different research priorities, and I won't go over every single one of them. But I still think, the biggest thing is understanding what's happening on the back end of ALK1 signaling. And so like, how is this complex affecting cellular behavior and how is that leading to AVM development? I think one big thing that really is needs to be prioritized is pregnancy and women's health. There's been very little research on that. I think it's a really important area. The other, I had the other one in mind and it kind of just slipped my mind. Yeah, so where the cellular behavior, and the last one that we kind of touched on at the last question is why certain AVMs bleed and others don't. And so, why do you get these differences in AVM phenotype or the way the AVM looks in one organ bed versus the other? And I guess following that, I'll give you one more. Is this idea of this loss of heterozygosity, so this ALK1 mosaicism. Is it that some patients have like a ton of AVMs because they just have more, fully mutated cells, or is some patients who have really large AVMs, that AVM just has a greater percentage of cells? So I think those are really kind of the main things that, at least on a basic science perspective, I think those are really the big areas of research.